Yes, all right, there are a lot of very, very important things that control your blood pressure. The good news is that we have many, many drugs that can affect these many, many things that affect blood pressure. So there's short-term, your neural control, your hormonal controls can do things immediately. Your long-term controls, your kidneys are actually going to do a lot to control your blood pressure in multiple ways. In fact, your kidneys work with both short and long-term blood pressure. So for example, if your kidneys detect that you have high blood pressure, the kidneys got to protect the kidneys. They've got a lot of vasculature, they've got a lot of capillary beds in there. So even in the short term, they can do things like if your blood pressure is low, they can decrease filtration. If your blood pressure is high, then can increase filtration. You can pee more or pee less. And that's actually going to alter your blood pressure because it alters this word, your blood volume. Blood volume. And you'll notice over here, this very, very complicated mechanism, the RAS system, the renin-angiotensin-aldosterone system, is also going to impact your blood volume via a number of different mechanisms. Yes, there's a reason I'm saying it like that. We do have to go into quite a bit of detail about the renin-angiotensin-aldosterone system, RAS, because I'm going to keep bringing up RAS over and over and over. And part of the reason it's going to keep coming up is because we have a variety of organs involved and a variety of systems involved. It starts with, and this is showing you low blood pressure. It starts with low blood pressure. You have low blood pressure for some reason. The first thing you do is your kidneys detect that low blood pressure and they release the hormone renin, or renin, depending on who you talk to. So the kidneys are actually endocrinal, they actually are hormonal organs. They release two major hormones. They release erythropoietin, which you learned about in endocrine lecture, and they release renin. And they're both very, very important for blood. So it turns out your kidneys and blood, very, very much connected. So in this case, they're releasing renin. Now your liver, if I could, I would have a little picture of a liver over here. Your liver creates a protein called angiotensinogen. And in the presence of angiotensinogen and renin, angiotensinogen is converted into angiotensin 1. If you have this enzyme floating around, ACE, angiotensin converting enzyme, that will automatically be converted into angiotensin 2. Angiotensin 2 is going to stimulate a number of systems. The one we're going to pay the most attention to right now is it's going to stimulate the adrenal cortex to release aldosterone. Do you guys remember what aldosterone did? Yep. I know you learned it. So it's the adrenal cortex, salt, sugar, sex. It's the salts. What about salt? Blood pressure, absolutely. So aldosterone stimulates, we're back to the kidneys. We came right back around. This stimulates the kidneys to retain salt and actually excrete potassium. And when you retain salt, you therefore retain water. Water goes to where salt goes. So aldosterone is going to cause sodium reabsorption and thus water reabsorption. And that's going to increase your blood volume or keep your blood volume high. And again, blood volume here, blood volume. And that is going to keep your blood pressure higher. Now, I'll also point out there's a couple of other things going on. You've got vasoconstriction. You've got increased water intake. You actually have a behavioral intervention for low blood pressure. You will drink more water when you have low blood pressure. That is the thirst mechanism, in fact. It's also going to stimulate antidiuretic hormone. Does anybody remember where ADH was from? The pituitary, and you're actually right, the hypothalamus synthesizes antidiuretic hormone and then it's released by the posterior pituitary gland. Excellent. And that is also going to cause increased water reabsorption in the kidneys. So obviously this is going to come back when we get to those kidneys, right? We're going to see this when we get to the renal system again. And that was the renin-angiotensin-aldosterone system, which is RAAS, S for system. Okay, blood flow through the body. Again, tissue perfusion means that there is blood in those blood vessels, which means that gas and nutrient exchange can occur. And of course, the whole purpose of this is to deliver oxygen, to remove carbon dioxide, any other metabolic wastes that are around, gas exchange in the lungs for that pulmonary circuit, absorption of nutrients in the digestive tract. So again, when we see our digestive system in a couple of weeks, we will be talking extensively about the vasculature of the digestive system. And urine formation, those kidneys are going to be very vascular, which is why you see them so much in blood pressure control. And again, rate of flow is, or should be is a better phrase here, precisely right amount to provide proper function. We're actually going to have a lot of mechanisms. If your body does not need as much blood, we are not going to send as much blood to it. At least that's the idea in a fully functioning cardiovascular system. And of course, when we get to patho, it's not going to be functioning properly anymore. Now this is just showing a diagram of volumes of blood in different areas. Again, I don't ever make you responsible for numbers. I just want to show you the overall concept here is that as you start to, for example, exercise, your oxygen and nutrient demands increase. Therefore, your circulation increases. So your blood volume didn't do anything to change, but your blood flow went up because your heart rate went up, right? So you're getting more blood to these areas over and over again in a shorter time span. So especially your blood volume delivered to your skeletal muscles increases considerably. So for most of these, you're going to see an increase. With your abdomen, you decrease your blood flow. Remember, fight or flight and rest and digest. You do not need to be digesting food while you're working out. So you will have less perfusion in your abdomen. You will have less blood flow there. And that's really all that this chart is getting at. Increased oxygen demands means increased perfusion, increased heart rate, increased cardiac output to increase that volume reaching the places that need it. Question? No? No. Okay. You can think about it for a second. You're allowed. Same volume of blood, just circulating a lot more. Again, so this ties back into our idea of cross-sectional area. The aorta is going to have very, very little cross-sectional area. The vena cava is going to have very, very little cross-sectional area. Even though they are the largest vessels, there's only one of these and only 2 vena cavae. So they're relatively small, right, in cross-sectional area. Whereas as these blood vessels split and go through many generations of splitting until they're smaller and smaller, there are more and more and more of them. So your total cross-sectional area of the vascular bed of your capillaries, this is saying it's 4,000 centimeters squared is our cross-sectional area of our capillary beds. And again, we'll look up some images later to really solidify this concept that your capillary beds are everywhere. There's so many of them that it doesn't matter that they're tiny. They're just massive in area. And because that area increases, the velocity decreases. And this is an important concept for you. It slows down across those capillary beds, and it never really quite picks up speed on the venous end. That's an important concept because that speed and that blood pressure that we saw earlier, those are going to cause blood to move out of or into capillary beds. It's going to be really important to you in a minute. So keep this in mind, that it slows down over this broad area. So think of it this way. You've got a waterfall over here. As it ejects from your heart, it's as powerful and fast as that is going to get, right? That's your waterfall area of your vascular system, cardiovascular system. When you get to this wider surface areas, it's no longer a waterfall. We've gone out to a plain. The water, that same volume of water that was in that sort of narrow waterfall, is now out across a broad plain. Now, if water is moving across a broad plain, is it going to be fast or slow? Probably slow. And then even if we collect that into a small river on the opposite side, is that going to be a fast-moving river or a slow-moving river? A slow-moving river on the other side. So do want to keep that in mind. And we've already conveyed this, vasodilators, vasoconstrictors. This is going to decrease the blood pressure in that immediate area. This is going to increase the blood pressure in that immediate area. We've already talked about some of the things that are going to affect this. The sympathetic nervous system, or the parasympathetic nervous system, controlling this decreased sympathetic tone, decreased release of epinephrine, norepinephrine is going to cause vasodilation, whereas more is going to cause vasoconstriction. We're also going to have a few other hormones and substances. So for example, nitric oxide, the cells of your blood vessels themselves create nitric oxide. Who's heard of nitric oxide before? Or its very, very close cousin, nitrous oxide. Yeah, what's up with it? Tell me about it. So using nitrous to like boost, probably. That's not the one I'm getting at right now. It's probably a different thing entirely.
Too Fast and Furious. I'm like, I don't know if I'm supposed to use this.
Like Fast and Furious. Yeah, it's boosted with nitrous. Whoa! I have no idea if that's what cars use to go really, really fast. Dentist office? Anyone?
Laughing gas.
Laughing gas, yeah. So they're both vasodilators, but the nitric oxide is the one produced by your blood vessels to cause vasodilation. So they're controlling their own blood pressure in very many ways. Atrial natriuretic peptide, ANP, is actually released by the atria of your heart. That's a hormone that's actually going to decrease blood pressure. Most of your hormones are going to increase it, but your heart hormones are going to decrease it. So when your atria of your heart notice that they're too stretched out, they'll release ANP, atrial natriuretic peptide, to decrease that blood pressure, to decrease that dilation. Yeah?
And how is that different than BNP?
They're very similar. BNP is released by the ventricles.
Oh.
And I don't know why it starts with a B. I wouldn't have named it BNP. Okay, down here, other things that cause vasoconstriction. You've already seen angiotensin II from that RAS pathway causing constriction. Antidiuretic hormone can cause some vasoconstriction, epinephrine and norepinephrine. Cool, right? So many things are affecting this. It's a lot of different aspects of it. Okay, mean arterial pressure. I think we're good there. Metabolic, myogenic. There's a couple of other things your blood vessels can do. Angiogenesis is the capacity of blood vessels to grow according to need. Angiogenesis is a normal physiological thing. If you are injured, let's say you break a bone, you're going to want to form a lot of blood vessels in that area to serve the metabolic needs of that bone as it heals, for example. It can also be part of a pathological process. If you are a tumor and your job is to just grow, you're going to need increased vascular supply if you're going to succeed at growing very far. Because, I mean, think of just the width of that aorta wall. It was kind of, or not, yeah, the wall of the aorta. It was thin to grow, you know, just to our eyeballs. That was a thin area. But that needs its own vascular supply. It's thick enough that simple diffusion of nutrients from nearby blood vessels or from within that blood vessel is not enough to supply it with oxygen and nutrients. So if you're a tumor and you're getting bigger and bigger and bigger, you're going to use angiogenesis. You're going to develop the chemical signals that stimulate angiogenesis to supply you with blood vessels. That's why if you watch like some kind of medical drama and they're talking about removing a tumor and they're like, no, the blood vessels, it's too dangerous, they're going to bleed out. Have you ever seen? There's always an episode like that somewhere. It's because it formed its own blood vessels. That's actually relatively medically accurate. Question there and then question here.
I was just going to say, yeah, because I had a bone tumor, and so when the doctor went in, he was really worried about that because it was the bloodiest thing.
Yeah.
So many, he had to end up going in twice.
Mm-hmm.
Way more than he expected.
Yeah.
But, yeah.
So a bone tumor that was very vascular. Yeah, absolutely. Ouch. That's no good. Yeah. Yeah, you could lose a lot of blood if you just went in and removed the whole thing. You're taking a whole lot of blood with you. So you had a question, and then we'll go back to you.
So with chemo, when you have cancer, like a tumor, and the chemo, is it attacking the blood vessels as well?
So not necessarily chemo, but we do have some drugs that specifically inhibit angiogenesis.
So is that why, like, my aunt had chemo or cancer when she was 15, and yesterday she found out she had 30% of her heart function right now.
Ouch.
So due to chemo.
Okay.
So is that what happened? Like the blood vessels, and then it also tired the heart and all of that?
Probably, yeah. I mean, I'm sure it was a lot of factors going into chemo affecting her heart health long term. Probably destruction, potentially, of the heart wall itself. Any radiation that would have if affected bone marrow would have affected blood cell production. There's a lot of things. Yeah, absolutely. So yeah, we do have drugs that can stop angiogenesis. So if you're taking a chemo drug to stop angiogenesis, what's the side effect of that going to be? Maybe not low blood pressure, but what happens if you break a bone or get a bruise? Yeah, poor healing. And then we are a high altitude area. We're in Denver, Colorado at 5280 feet right now. That means we have more blood vessels than people at sea level, which is pretty cool. Which is why when we go to sea, one reason why when we go to sea level, we just go, wee, I'm on a run, it's awesome. And if you've never gone on a run while on vacation at sea level, I recommend that you do, because it is awesome.
It is great.
Yeah. I went for, I think, 2 or 3 runs while I was at my mom's place in Florida over break, and it was just like, la la la, la la la, la la la, the whole time. Just fantastic. Yeah.
So, when I went in for my surgery, they went and did, where they go in and block all the blood vessels to where it is?
Yeah.
So why would they not use a drug? Why would they go do that?
That's a good question. Why would they not just do a surgical intervention instead of a medical intervention to block off angiogenesis? I imagine even if you're cauterizing a whole bunch of blood vessels to a tumor to kill it off, it can still produce new blood vessels after that. That's what I'm imagining. I would still ask like some person who works with chemo patients to talk about that a little bit more. But that's my suspicion, is it can just keep doing it. Yeah. Which is a pretty intense mutation, right? Like it takes some mutation for a cell that has never had the chemical component of angiogenesis before to develop that component of angiogenesis. Like if you're a skin cell, you're avascular, you don't really have that skill, right? I might be wrong about that. There's some cell biologists out there like, no, that's not true, but I don't know. It's a series of mutations for cancer, right? It's a really complicated cascade for that to happen successfully. Okay, so that was our autoregulation, our long-term, our short-term control of blood pressure and blood flow. And now we're going to get into why did we do this in the first place? For gas exchange. And of course, this concept is going to come back in our respiratory lecture. Things are just going down their concentration gradients. If you're in a capillary bed, it's a single layer of simple squamous epithelium. It's an endothelium. And that is very small because oxygen is going to diffuse down its concentration from high inside of the arterial end and low out in the tissues. And then your tissues are going to be very high in carbon dioxide. So carbon dioxide is going to diffuse from your tissues into your capillary bed in that tissue. Other things like lipid soluble molecules are going to diffuse directly through. Water soluble solutes are going to go through clefts and fenestrations in which means that we're going to need at least fenestrations when we get to the digestive system, right? Because we're going to have to get our proteins, fats, carbohydrates from our intestinal lumen into our capillary beds of the digestive system. And things like proteins, pinocytosis, or things like that are going to be how we get larger molecules through that lumen. I think that's all I got there. There's that fenestrated capillary bed. There's its basement membrane. There's pinocytosis happening. And again, even with some of those fenestrations, there's still going to be a membranous barrier there. It's not going to be quite as much of a barrier as an entire cell, but it's still going to be selective. We're coming up on the other huge concept. RAS was one of them, but bulk flow is the horse. This is the horse. And this horse, I'm going to beat this horse, and I'm going to beat this horse to death, and this horse is going to die, and I'm going to keep beating a dead horse for the rest of your education here that you work with me. And you're going to keep seeing it in other courses and related to other concepts. I know it's not really pretty that I'm saying dead horse over and over again. But this is the horse. It's never, ever, ever going away. So I'll do it once straight through for the recording, then we'll do it on the board, and we'll do it like 5 more times after that. So this is, consider this to be a capillary bed. This is a simplified capillary bed. The arterial end is over here. The venous end is over here. And one of the first concepts we're going to tie into this is blood pressure and velocity. Remember that from the arteries, you've got very high blood pressure and velocity. And on the venous end, you've got very low pressure and low velocity. So far, so good? Okay. Does anybody remember the plasma protein that exerts osmotic force? Albumin. Excellent. Remember how much I emphasized that then, because it's coming back today. So straight through, simplest version, on the arterial end, you have higher hydrostatic pressure than osmotic pressure. There's a certain amount of albumin in here, and that is pulling water back into the capillary. But you have a whole lot of hydrostatic pressure created by blood pressure, by the force and velocity just of fluid flowing from that heart. It's pushing against the water, the walls of the capillary, and that water is being pushed out. Therefore, on the capillary end, net flow, more water is flowing out of the capillary bed than is flowing back in. So far, so good? Almost? Again, we will repeat this about 1,000 times. We cross the capillary bed, and as we cross the wide expanse of the capillary bed and head towards the venous end, the velocity decreases, the blood pressure decreases, therefore the hydrostatic pressure decreases. But we still have albumin. We still have plasma proteins exerting plasma force, so there's still plasma proteins pulling fluid back in to the capillary bed. And on the venous end, that capillary osmotic pressure is higher than the capillary hydrostatic pressure. Therefore, more fluid flows into the capillary bed on the venous end than flows out of the capillary bed on the venous end. I'll repeat it one more time for the recording, and then we'll go to the board. Here's the arterial end. Here is the hydrostatic pressure of the capillary bed. And they have some numbers. I won't have you memorize the number, but they are representing this as 35 millimeters of mercury of pressure, pushing fluid out of the capillary bed. And then your osmotic pressure, caused by the pull of albumin and other plasma proteins, is equal to about 26 millimeters of mercury. So that's not as powerful as the push. The hydrostatic pressure is greater than the osmotic pressure of the capillary. Therefore, fluid flows out at the arterial end. On the venous end, our hydrostatic pressure of the capillary has decreased. We went over that wide surface area. There's no more blood pressure, or at least not very high blood pressure. There's not very high blood velocity. So our push of hydrostatic pressure is only about 17 millimeters of mercury, whereas our osmotic pressure exerted by albumin and other proteins is still 26 millimeters of mercury. Because there's more osmotic pressure than hydrostatic pressure, more fluid moves back in to the capillary bed at the venous end. And one more thing before I wrap this up. I've so far only been considering the pressures inside of the capillary bed. We also have to consider that there's fluid and stuff and pressures outside of the capillary bed in the tissues. They're going to call it interstitial fluid. The fluid between your cells has pressures on it, and it has stuff in it. So you have a little bit of hydrostatic pressure, pretty much nothing at this point. They're calling it 0 millimeters of mercury, of pressure of just the water between your cells. And there's a certain amount of stuff, but it's not a lot. So osmotic pressure of the interstitial fluid is only about 1 millimeter of mercury. It's very low. And it's about the same on the venous and arterial ends, 0 and 1. So they're not as big pressure-wise as your capillary hydrostatic and osmotic pressures. But we still need to consider them. And one reason is that if you have more fluid in the interstitium than you should, what's that called? Edema. And what's one thing, if somebody's just got minor edema or they're concerned about edema, what's something that we do for them? Salt restriction. What else, though? Diuretics, again, more medical. What about something physical that we can do for them? Elevation is an option. What else? Compression. TED hose, right? What of these pressures is TED hose going to influence? The interstitial fluid, is it the hydrostatic or osmotic pressure of the interstitium? Hydrostatic. So TED hose are going to increase interstitial hydrostatic pressure to try to get more fluid back into the capillary bed. So that's why that's important. And again, just to review this idea of osmosis, this is kind of a classic experiment that we've been able to do before in different lab environments. This would be part of like a Gen Bio class, if you guys had a Gen Bio class that you took, which you don't really. So this is demonstrating hydrostatic pressure on this side. If you took a piston and you pushed it, and there's a semi-permeable membrane right here, eventually we would be able to push some of that fluid across the semi-permeable membrane. That's hydrostatic pressure. I think this experiment, which I have done in different contexts, is much more fun. If you have a semi-permeable membrane here, you put a solute in one side, for example, albumin. Let's say there's albumin on this side, and the albumin cannot cross this membrane, but water can. Water is a lot smaller molecularly than albumin is. If you just leave this here, you don't have to do anything to it, eventually there will be more water on this side than there will be on this side, because water goes to stuff, absolutely. It's going to equalize the concentration. So if at the beginning of the experiment you took a sample here and a sample here, the concentration would be, 1 albumin versus 5 albumin for just these sample sizes, right? Whereas if you took a sample here at the end of this experiment and a sample here, you've got 3 albumin and 3 albumin. It's equalized. It's the water that is changed. It is the water that was pulled by the stuff. And I know I'm being really repetitive, but I find people need the repetition for this one.